Good evening, students. Welcome back to the class. And today we are going to discuss some of the important questions related to the topic cell biology, genetics, and blood. So let us know about this part 1 here. And the first question under this topic is, in which one of the following antibody formation takes place? RBCs, blood platelets, blood plasma cells, Down's membrane. Okay, let us look at the right answer here. The right answer is option C, blood plasma cells. So here in the blood plasma cells, antibodies will get synthesized. So when we are looking at the volume of the blood, the total blood that we have in our body, approximately about 55 to 60% of this blood consists of blood plasma cells. And these plasma cells includes antibodies and some other lysosome. All of these components are basically present, embedded there in the plasma cells. And all of these serves together to destroy the bacteria, virus, toxic substances, or any other foreign substance that enters our bloodstream from outside usually, or if there is any damage in the tissues. So usually the antibodies are generally synthesized in our body to fight off those damaged cells. Okay, usually the foreign substances, if it is there in our body. Basically, these antibodies are required to our body to enhance our immunity system, to prevent our body from various diseases. Okay, moving to the next question now. pH of human blood is 7.4, 6.2, 9, 7.5 to 8. Okay, let us look at the right answer. The right answer is option A, 7.4. So 7.4 is a normal pH level of our blood. So what is this pH? pH is nothing but potence of hydrogen. Okay, potence of hydrogen in a solution. That is to know whether the solution that we have, whether it is acidic or basic in nature. So usually here in the pH scale, that tells us whether the solution that we have, the mixture that we have, whether it is acidic or basic in nature. When we are looking at the blood here, so here we have blood is slightly alkaline in nature. That is, it is quite basic in nature. Okay, so this is where it comes in between 7 and 8. That is where the pH value of the blood lies. So when we are looking at this pH scale here, we will get to know that hydrochloric acid it is highly acidic, whereas dry cleaners that we will use at home, okay, in drainage. So there, that is the one which is strong base. Just for example, NaOH, KOH, okay, sodium hydroxide, potassium hydroxide, all these are strong bases. Whereas H2SO4, sulfuric acid and hydrochloric acid, all these are strong acids. Our blood is approximately, it is neutral in nature. Moving to the next question now. Which one of the following is not a constituent of cell membrane? Cholesterol, glycolipids, proline, phospholipids. Okay, let us look at the right answer. The right answer is option C, proline. Okay, proline is it is not a constituent of cell membrane. It is not present in cell membrane that we have in our body. We know that our body is made up of trillion number of cells. So here in all the cells, the cell membrane, which is the outer covering of the cell, there will be no presence of the proline. But apart from that, cholesterol, glycolipids, phospholipids, all these are present there in our cell membrane, because basically it is made out of lipids as well as proteins. Okay, our cell membrane is made out of lipids and proteins. Here glycolipids, phospholipids, cholesterol, which is nothing but sterol. It is one of a sterol. Okay, sterol, sterol. So, which is a constituent of our cell membrane. But there is no presence of proline in our cell membrane. It is an amino acid. Proline is an amino acid among the rest of the 20 DNA encoded amino acids. Here proline is one among them. And L-proline is the one which is used as osmoprotectant. Okay, here osmoprotectant, why do we use this as an osmoprotectant? Because when there is highly extreme stressful conditions, stressful osmotic stress there in an individual, usually this proline is the one which is used in the pharmaceutical as well as biotechnological applications, that is in the medicines, because it will reduce this extreme osmotic stress. And also it helps us to survive, helps an organism to survive. So it has a major application there in pharmaceutical industries. Move to the next question. Which of the following contain enzymes for cellular respiration? Dictyosomes, endoplasmic reticulum, lysosomes, mitochondria. Okay, let us look at the answer here. The right answer is option D, mitochondria. So mitochondria is the one which contains specific enzymes, and these enzymes which are present in the mitochondria is responsible for cellular respiration. But what do you mean by the cellular respiration? Because the actual respiration in our body, it takes place in all the cells that we have. Our body has trillion number of cells in all the different parts of our body. So this is a place where actual respiration takes place, right? In all the parts of our body. Breathing only happens in between our nostrils and the lungs. That process we call it as breathing. But the actual respiration takes place in the cells, that we call it as cellular respiration. The food that we have, it will get digested in the digestive system. So once it gets digested, the nutrients which is present in the food is then transported to different parts of our body where there is a presence of cells. So once this nutrient enter our cell, it has to be then broken down into simple substances, right? So while it is broken down, energy is also released. Why do we require the food? To get the energy out of it, right? That is what we call it as cellular respiration. The respiration process which will take place in the cell, specifically there in mitochondria of the cell. Okay, that is the reason we also call this mitochondria as powerhouse of the cell, because it releases energy molecule in the form of ATP. Moving to the next question now. The phenomenon of genetic mutation cannot occur in DNA, RNA, chromosome, ribosome. Okay, let us look at the right answer. The right answer is option D, ribosomes. Okay, ribosome is a site where genetic mutation does not occur. But apart from that, the rest of the option, if you are looking at this here, DNA, RNA, chromosome, these are the sites of genetic mutation to occur. Okay, basically it happens there in DNA and RNA, whereas this chromosome is a part of our DNA. Okay, chromosome is a part of DNA. We can consider this where actually the genetic mutation occurs. But when we are looking at this option, ribosome, ribosome is also known as protein synthesizing factories. Okay, it is just a part of our cell here. It is one of a component of our cell where the proteins are synthesized in our body. Okay, it is a site of protein synthesis, whereas DNA, RNA, and chromosome, they are the site of genetic mutation. So genetic mutation, it has to happen so that we can transfer the characters from one generation to the next, right? Moving to the next question. The function of nucleolus is the synthesis of DNA, mRNA, rRNA, tRNA. Okay, let us look at the right answer. The right answer is option C, rRNA. Okay, so here rRNA is the one which is synthesized here in the nucleolus of the cell. Okay, so if this is the cell, at the center of the cell, there will be presence of nucleus. And again, at the mid part of this nucleus, there is a presence of nucleolus. Okay, this is a nucleolus where this rRNA is synthesized in our body. Okay, rRNA synthesis takes place here in the nucleolus. But when we are looking at the rest of it, so this rRNA is also known as ribosomal RNA. Okay, ribosomal rRNA. Okay, ribosomal RNA, ribonucleic acid. So when we are looking at mRNA, which is nothing but messenger RNA. Okay, messenger RNA. So that is the one which is required to convey the genetic information from DNA to ribosome. So here the major function of this mRNA is to transfer the genetic information from DNA to the ribosomes. Whereas this tRNA, what is the function of this tRNA? Transfer RNA. Okay, transfer ribonucleic acid. So here we have the full form, right? So it is one of a type of RNA molecule itself. It is required to decode the messenger RNA, that is mRNA. Whatever the sequence that it is carrying, the genetic information, to decode this information, we require tRNA. Okay, so it will decode and it will then convert it into protein. Okay, so then when we are looking at the last option, that is DNA, deoxyribonucleic acid. So it is one of a molecule that carries the genetic material which is required, which provides us the information regarding the growth, development, functioning, and also reproduction in an individual. Okay, moving to the next question now. During which stage of mitosis does cytokinesis usually occur in animals? Prophase, metaphase, anaphase, telophase. Okay, let us look at the right answer. The right answer is option D, telophase. Okay, we know that all these are the different stages of mitosis. Mitosis is nothing but the cell division, right? If the cell has to divide, it has to undergo this process of mitosis. So within which there is a presence of 4 different stages: prophase, metaphase, anaphase, and telophase. So here in this telophase, this is the final stage. Okay, once the DNA material, everything is ready for the cell to undergo division. So here telophase is the last final stage of the cell cycle, during which cytoplasm will usually, cytokinesis will occur. Cytokinesis, which is nothing but division of cytoplasm. If we have a mother cell just like this, now it had made all the required materials for its division. Okay, there will be formation of furrow like this. Okay, it will form the furrow at the center of the cell. okay, which will deepen as and when it reaches the next stage. So finally, it will form two different daughter cells. Okay, two different cells, but both of these two different daughter cells will be of the same identical character to that of the parent. Okay, this process of cell division, that is cytokinesis, where there will be division of the cytoplasm, that occurs in telophase of cell cycle. Next question. Carbohydrates are stored in plants and animals in the form of cellulose and glucose respectively, starch and glycogen respectively, starch and glucose respectively, cellulose and glycogen respectively. Okay, let us look at the right answer here. The right answer is option B, starch and glycogen respectively. Okay, that is here in plants, the carbohydrates, they usually consist of complex sugar molecule, that is complex glucose, right? It has to be then further broken down to release the energy, because we know that carbohydrate is energy giving food, right? So this carbohydrate is a form of glucose, and this glucose is a simple sugar, which is nothing but monosaccharide. That is only one. Saccharide is nothing but sugar unit. Okay, monosaccharide. So it consists of, that is glucose consists of only one sugar unit within that. Okay, so this glucose here in the plants, it is stored in the form of starch. Similarly, in case of animals, it is stored in the form of glycogen. Okay, in the plants, it will be in the form of starch, whereas in animals, it will be in the form of glycogen. And we know that glucose, even though since it is a simple sugar, that is the one which has a major part in the metabolic pathways, right? The major part, because it is the one which will release the energy required for all the activities that we do in our daily life, right? So in carbohydrate basically stored in case of plants as a starch, and in animals as glycogen. Next question. Which of the following is related to glycosylation of protein? Endoplasmic reticulum, peroxisome, lysosome, mitochondria. Okay, let us look at the right answer here. The right answer here is option A, endoplasmic reticulum. Okay, because here in endoplasmic reticulum, glycosylation, okay, this glycosylation of protein is the major part, and that actually happens here in our endoplasmic reticulum of our cell. Okay, so when we are looking at most of the proteins, almost all the proteins which are synthesized here in endoplasmic reticulum, they are bound with ribosomes. If they are bounded with the ribosomes, then we can call the same as rough endoplasmic reticulum. If it is not with ribosomes, then it is smooth endoplasmic reticulum, right? So since these proteins, ribosomes are bounded here to these proteins, we can consider the same as rough endoplasmic reticulum. And these are the one which are then converted into glycoproteins. Okay, glycoproteins, and this further undergoes glycosylation that occurs in the rough endoplasmic reticulum. Glycosylation actually occurs here in rough endoplasmic reticulum, that you have to make a note of this. Clear? What do you mean by glycosylation now? Glycosylation is nothing but it is a bond, okay, bonding of sugar residues to another organic compound. So when there is a bonding between sugar residues, different sugar residues like glucose, fructose, galactose, sucrose, lactose, maltose, all these are sugar residues. If it is combining with organic compound, this process we call it as glycosylation. Adding of glucose units to organic compound, that process is glycosylation. That actually occurs in our endoplasmic reticulum, specifically in rough endoplasmic reticulum, since there is a presence of ribosomes on it, okay, which will make the surface rough, okay, rough comparatively to that of smooth endoplasmic reticulum. Moving to the next question. Which one among the following nutrients is a structural component of the cell wall of plants? Manganese, potassium, phosphorus, calcium. Okay, let us look at the right answer. The right answer is option D, calcium. Okay, calcium is one of the structural component of the cell wall in case of plants. Of course, even calcium is present even in animals' body too, even in our body too. But it is one of the structural component of the plants compared to all of this. Okay, because this calcium is the one that activates enzymes. Okay, these enzyme, it is a structural component of the cell wall. Calcium, if there is a presence of calcium in the cell wall, that can activate enzymes present there, okay, which will provide the strength required for the cell wall in case of plants. So that is the one which actually the enzyme present here, once it gets activated here because of the presence of calcium, so it will get activated, and it is the one which will influence the water movement in cells. And definitely this water movement is necessary in case of plants because it has to grow, it has to divide. For all this reason, water has to move there in and out of the cell through a semi-permeable membrane, right? But when we are looking at this next rest of the options, phosphorus. Phosphorus helps in energy flow there in case of plants. Okay, manganese is the one that is involved in enzyme synthesis. So enzymes are necessary because it is the one which will carry out most of the metabolic activities, even in case of animals too, right? So this enzyme synthesis, manganese is the one of a mineral which is involved. And phosphorus, that makes the backbone of genetic materials like DNA and RNA. Okay, it provides the backbone structure for this DNA and RNA. We know that it is a double helix model, DNA, right? Double-stranded, so double helix model, to provide the structure to DNA and RNA, there requires this phosphorus because it acts like a skeleton there. Moving to the next question now. Which of the following blood group is called universal donor? A positive, B positive, O positive, AB positive. Okay, let us look at the right answer. The right answer is option C, O positive. O positive is considered as universal donor. That is a person who has this blood group O positive can donate blood to anyone else, for everyone. Okay, so with different blood groups, because here a person with O positive blood group, they doesn't possess any antigen. Okay, in their blood actually the antigens are not actually present, and the antigens which can usually elicit antibody reactions in the acceptor. For example, if I am accepting the blood from O positive person, so usually this O positive blood doesn't contain any antigen so that it can elicit the antibody that is present in my body. So usually doesn't react with each other. Okay, there will be no any opposite reaction that takes place in my body. So I can receive this O positive blood. Okay, so that is the reason O positive is known as the blood group O positive is universal donor, whereas this AB positive, okay, AB positive is considered as universal acceptor. That is, they can receive the blood from everyone else. Okay, they can easily accept it, all the different kinds of blood. So we can even provide this A positive, B positive, O positive. All the different blood groups can be provided to AB positive person. Moving to the next question. Cells which take part in secondary growth are named as phloem, xylem, cambium, medullary ray. Okay, let us look at the right answer. The right answer is option C, cambium. Okay, cambium is the one which takes part in secondary growth in case of plants. So what is the secondary growth? So usually the dicotyledonous plants, okay, most of this dicotyledonous plants, they exhibit increase in girth, right? Increase in girth, that is what we call it as secondary growth. So for example, if this is a part of a tree, okay, the trunk of the tree, usually it increases when we look at every year of the same tree. So it will usually increase in the girth of this tree. So when there is an increase in the girth, it's all because of the occurrence of the secondary growth. Okay, for the plant to undergo secondary growth, if they have to undergo secondary growth, definitely they have to possess some of the tissues in their body, right? So here the tissue which is responsible for plant growth, that is increasing girth, that is cambium. Cambium is the one which is usually present in the plants, specifically there in dicot plants, which helps in increasing girth, that is to help them undergo secondary growth. Okay, usually when we are looking at this cambium, this tissue is of two different types, two lateral meristems. Okay, so the secondary growth or the two lateral meristems. The first here is vascular cambium and cork cambium. So vascular cambium is the one which is responsible for the increase the diameter of stems and roots. Okay, so vascular cambium is the one which helps the plant to increase its diameter of the stems as well as the roots. Whereas this cork cambium, okay, cork cambium, it is the one which produces bark of the tree. Okay, this bark of the tree, it is all produced by the cork cambium, this tissue. Clear? Moving to the next question now. Which of the following does not have any enzymes in its cell? Algae, virus, lichen, bacteria. Okay, let us look at the right answer. The right answer is option B, virus. Virus is the one that does not consist any of the enzymes in its cell because it is not a cell at all. Okay, virus, we cannot call it as a cell because no cell is present in its body. So this genetic material, whether it can be DNA or RNA, this genetic material, it is freely embedded, okay, freely suspended there in this virus. Okay, encapsulated on outside with a protein capsid. So protein capsid, there will be outer covering of this virus. So protein capsid will be here. So this is how it will be. So if you are considering bacteriophage, okay, the outer protein covering, that is this part we call it as head of this virus. Okay, so outer covering of this head is actually made out of proteins. Okay, that part we call it as a capsid, which is a proteinaceous in nature. Usually in this part, inside this, there will be presence of genetic material. And once this virus reaches the host body, host cell, whether it can be plants or the animals or even human beings, right? So once it enters host cells, there it can inject all the genetic material so that it can transform the genetic material to the host body. Okay. Moving to the next question now. Which of the following substances, if introduced in the bloodstream, would cause coagulation at the site of its introduction? Fibrinogen, prothrombin, heparin, thromboplastin. Okay, let us look at the right answer. The right answer is option D, thromboplastin. Okay, thromboplastin, when it is introduced into body, usually it will start blood coagulation at the site of its introduction. Okay, so this thromboplastin is actually necessary to convert this prothrombin into thrombin. For this conversion, of prothrombin into thrombin, thromboplastin is necessary. So this prothrombin, when we are converting into thrombin, thrombin is the one which actually takes part. It aids coagulation process in case of animals. Okay, so to convert, to coagulate the blood, that is the clotting factor, right? Coagulate the blood, thromboplastin is the one which acts as a catalyst, because it is the one which will convert prothrombin into thrombin. Okay, so when we are looking at this fibrinogen, fibrinogen is one of a glycoprotein. Okay, it is a glycoprotein which is basically present in all the vertebrates, which is necessary for blood clot formation if there is an accident. Okay, blood has to clot. So for that, fibrinogen is as a major activity, major responsibility there. And when we are looking at this heparin, it acts as anticoagulant. That is, it will suppress the clotting factor. Okay, and when we are looking at this, so considering this question here, at the site of introduction, so wherever we introduce this thromboplastin here into our body, into animal's body, so it will usually convert all the prothrombin present there into thrombin. So when it is converted, definitely that site, that part will get coagulated. Blood clotting will happen. So when we are considering all the option here, thromboplastin is the one at the site of introduction, it will start clotting, because there it is the one which is converting the prothrombin into thrombin. Without conversion, okay, definitely not clot. So that is the reason thromboplastin, we can choose it here. Moving to the next question. Average lifespan of human RBC is 100 days, 90 days, 120 days, none of these. Okay, let us look at the right answer. The right answer is option C, 120 days. So this is the average lifespan of human RBC. Okay, the average lifespan of human RBC, red blood corpuscles, red blood cells that we have in our blood, one of a type of blood cells. It can survive, it can be alive, it can be active only for 120 days of its lifespan. Okay, this is a lifespan of red blood cells that we have. Next question. Certain parts of a plant can be bent easily without breaking. This flexibility in certain parts like leaf and stem can be attributed to the abundance of parenchyma, collenchyma, sclerenchyma, xylem and phloem. Okay, let us look at the right answer. The right answer is option B, collenchyma. Because of the presence of collenchyma in case of plant can bend easily. There is a flexibility found in plants. Okay, all of these are different tissues which are found in plants, plant tissues. Okay, meristematic tissue basically. So under this meristematic tissue here we have parenchyma, collenchyma, sclerenchyma, permanent tissues. Okay, this collenchyma which is present in the plants, that is the one which will provide malleability as well as flexibility. That is, it can bend easily. Okay, plant can bend easily when there is a blow of the wind. When the wind blows with high speed, definitely it will bend. But again, it will come back to its original position, right? It will not break immediately. It's all because of presence of collenchyma there in case of plants. It provides the plant flexibility. Okay, parenchyma which is present in the plant, it is considered as soft tissue which are present in the inner part of the plant. Okay, which makes most of the cell in case of plants. And sclerenchyma is a dead tissue, and it is the one which makes up fiber and also provide the rigidity to the cell. Okay, if there is a cell like this, the outer part of this, it has to provide a rigidity so that easily the plant can bend and can come back to its original position, because there is a presence of sclerenchyma too. It is a dead tissue and provides the rigidity, strength to the cells. Okay. And when we are looking at the xylem and phloem, these are vascular tissues, okay, which is required for the conduction of food and water in case of plants. Moving to the next question. Blood and lymph are types of muscular tissue, epithelial tissue, connective tissue, permanent tissue. Okay, let us look at the right answer. The right answer is option C, connective tissue. Okay, both blood and lymph. So both of these are the types of connective tissue. Okay, connective tissues are named because of their special functioning like linking and supporting all the tissues as well as the organs in our body, right? We have different organs, different tissues in our body. If it has to be connected with each other, definitely we require this connective tissue. We know that blood is the one as well as lymph is the one which will flow throughout our body, right? This both will usually flow throughout our body. While flowing throughout our body, it will also carry the different information, different particles, components from one part of our body to different parts, right? So it acts as a connective tissue. Okay, so here when we are looking at this connective tissue, they range from soft connective tissue to dense connective tissue. Some of it will be very soft and some of them will be very hard, and also it will be very dense, compactly arranged within each other. Okay, so here we can see some of the specialized types of the cells, that is the tissue connective tissue or cartilage, that is the soft part of the bone that we have. So in the external ear pinna that we have, external ear, we can bend this part of our ear easily, right? Because we cannot bend our elbow, the bone that we have in our arms or the wrist. But the bone that we have at the tip of the nose, we can bend it easily because there is a presence of cartilage. Okay, it is neither too hard nor too soft. Bone, adipose tissue. Adipose tissue is the one where the fat is usually stored. Blood as well as lymph. Okay, so all of lymph is a connective tissue again, just like a lymphatic system. Okay, it is one of a part of lymphatic system. So all of these are the parts of connective tissue. Moving to the next question. The main function of Golgi body is respiration, to begin cell division, to produce gastric juice, secretion. Okay, let us look at the right answer here. The right answer is option D, secretion. Okay, the main function of our Golgi body, okay, Golgi body, which is present in the cells, is to secrete. What it actually secretes? Usually proteins are made in endoplasmic reticulum. Okay, endoplasmic reticulum of our cell, it is the one which will make the proteins. Okay, and these endoplasmic, the proteins which are synthesized here, where there is a presence of transition vesicles. Okay, here at this side, there will be synthesis of transition vesicles, which will usually flow through the cytoplasm. And finally, it reaches Golgi bodies. That is from endoplasmic reticulum through cytoplasm, it flows and finally reaches our Golgi bodies. Okay, so now after the Golgi bodies, so once the Golgi body completes all its work, the vesicles, okay, the vesicles which are synthesized there, transition vesicles which are synthesized there in endoplasmic reticulum. So now here in the Golgi apparatus, these vesicles acts as a secretory vesicle. Okay, with the help of the secretory vesicles are created, and the same secretory vesicles are then synthesized and then released to the cytoplasm. Okay, basically there in endoplasmic reticulum, transition vesicles are synthesized and released to cytoplasm. This reaches Golgi bodies. Here in the Golgi bodies, vesicles, that is secretory vesicles, are created as well as synthesized and released to the cytoplasm. Okay, so this is a main function of the Golgi body. Golgi apparatus, it is to absorb, it is to synthesize as well as to secrete the vesicles. Okay, vesicles again has a major role in our body. So when we are looking at the rest of the part here, respiration. We know that respiration happens both in case of plants and animals, right? So during this respiration process, we will take in oxygen, we will leave out carbon dioxide. The same happens even in case of plants. Cell division. Cell division, it is one of a phase of cytokinesis. Cytokinesis means division of the cytoplasm of the cell, which happens through mitosis as well as meiosis process. Okay, these two are the types of cell division. And gastric juice. So gastric juice, it is basically released in the stomach and the other glands, small intestine. It is to release, which is required for digestion process. Okay, so this gastric juice has a major role in the digestion of the food. Moving to the next question now. The double helix model of Watson and Crick is known as cDNA, bDNA, zDNA, ddNA. Okay, let us look at the right answer. The right answer is option B, bDNA. Okay, there are different types of DNAs that we can see here, cDNA, bDNA, zDNA, as well as ddNA, right? So one of the secondary structure exhibited by the DNA is famous for Watson and Crick model, right? Watson and Crick model, it shows all the necessary character that a DNA has to exhibit. So it completely explains about DNA structure. Most well advanced design of the model. It was all designed by Watson and Crick, right? And it is most suitable for the explanation of DNA structure. And here this model says DNA is double helix. That is, there is a strand, two different strands which are present there in DNA. So this is how the strands run along with each other, right? Double helix model. And these two strands of polynucleotides and are anti-parallel. Okay, here we have one strand and here we have the second strand. Both of these two strands are they consist of polynucleotides. Nucleotides. Basically, here the nucleotides which are present here are adenine, guanine, thymine, as well as cytosine. Okay, adenine, guanine, thymine, cytosine. So all of these are the nucleotides which are present in this double helix of DNA. Okay, along with that, it also run anti-parallel. That is, one if it is running, one of the strand if it is running in this direction, one will move in this direction, opposite to each other. Okay, so but when we are looking at these options that we have here, bDNA is the one usually that possesses double helix right-handed structure. bDNA choose double helix. The strand of this is double helix as well as right-handed in structure. And also it consists of about 10 to 10.5 nucleotides per ton. Okay, 10 to 10.5 nucleotides per ton. That is from year to year. So this is one turn of the double helix model, right? So within this, there will be presence of 10 to 10.5 nucleotides. So here the nucleotides are adenine, guanine, thymine, as well as cytosine. And also here this bDNA says that contain major as well as minor groove. And major groove is wider one when we compare with that of the minor groove. So it satisfies all the double helix model which was given by Watson and Crick. Okay, but when we are looking at the rest of the options, cDNA. cDNA is the one which will usually consist of only 9 base pair per ton. Okay, that is in this part, there will be presence of only 9 base pairs and it is right-handed, whereas this zDNA, okay, zDNA is the one which consists of 12 base pairs and along with that it is left-handed, it turns like this. Okay, so that is the reason when we are considering all the options, your bDNA is the one which completely suits. Okay, so Watson-Crick model, so this bDNA double helix model which was given by Watson and Crick, it is also known as bDNA. Okay, move to the next question now. Lysosome arises from endoplasmic reticulum, Golgi apparatus, nucleus, mitochondria. Okay, let's look at the answer. Golgi apparatus is the one. Option B, Golgi apparatus is the one. Okay, lysosomal membrane. It is formed in endoplasmic reticulum. Okay, endoplasmic reticulum, it is the one which also synthesizes lysosomal membrane. So this lysosomal membrane is then modified as well as transferred to cis phase. Okay, if we know about the structure of this Golgi apparatus, this is how it will be, which form cisternae with each other, right? So this is how the phase of this Golgi apparatus will be. So this lysosomal membrane are then transformed as well as modified into cis phase of this Golgi apparatus. Okay, this is a part of this Golgi apparatus where further process takes place. Here in this Golgi apparatus, the further processes of this lysosomal membrane takes place and then finally releases lysosome. Okay, that is one of the major function of Golgi apparatus. Not only packing and dispatching the food from one part of the cell to another, it also helps in the release of lysosomes. Lysosomes are synthesized and secreted here through Golgi apparatus. That is one of the major function of this. Okay, moving to the next question. Antigen is a substance which lowers body temperature, destroys harmful bacteria, triggers the immune system, is used as an antidote to poison. Okay, let us look at the right answer. The right answer is option B, triggers the immune system. Okay, antigen is the one. It is a substance which are released. Okay, when the foreign bodies, if a foreign substance or any other virus, bacteria, if that enters our body to the bloodstream directly, so it is the one which can destroy our body, right? Which can cause diseases in our body. So it acts as a foreign substance. Now, when there is a presence of antigen in our blood, so this antigen is the one that can start triggering our immune system. So our immunity system will start activating. It will get activated and it will also activate the lymphocytes present here. Antigen is responsible for the activation of lymphocytes. Lymphocytes is a part of our immune system, which finally starts infecting, okay, destroying this infection, which is caused by the foreign particles. So our white blood cells will get activated and it can also start fighting against the foreign substances that has entered our bloodstream. Okay, so antigen is required to trigger our immune system. It will trigger our immune system. Once it is triggered, it will start releasing antibodies which can fight against the antigen. It logs the surface biochemical pathway of the antigen. Next question. The replication of DNA cell to undergo division. During the cell cycle, the DNA replicates in S phase, G1 phase, G2 phase, M phase. Okay, let us look at the answer. The right answer is option A, S phase. That is nothing but synthesis phase. Okay, so here in synthesis phase, so here you can see cell cycle. Cell, if it is ready to undergo division, so it passes into different stages of the cell cycle. Okay, here the first one that we have here is G1 phase. Okay, this is the first phase of cell cycle, which is then followed by synthesis phase. Okay, G1 is nothing but gap 1 phase, synthesis phase, where the DNA synthesis takes place. Okay, DNA replication or DNA synthesis will take place here in synthesis phase. That is the second stage of cell cycle, which then undergo G2 phase, gap 2 phase, then mitosis phase. So this is a place where it will divide to form two daughter cells. Okay, but here DNA is the one which is synthesized in S phase, synthesis phase of cell cycle. Next question, which of the following groups is present in animal cells? Mitochondria, cell membrane, cell wall, cytoplasm. Option B, chloroplast, cytoplasm, vacuole, nucleus. Option C, nucleus, cell membrane, mitochondria, cytoplasm. Option D, vacuole, cell membrane, nucleus, mitochondria. Okay, let us look at the right answer. The right answer is option C. So here they have given us about animal cell, right? So in the animal cell, there is a presence of nucleus, cell membrane, mitochondria, as well as cytoplasm. So all of these parts are present in case of animal cells. Okay, when we are looking at the rest of the options. So cell wall is not present in animal cells. It is present only in case of plant cells. Chloroplast, which is required for photosynthesis because it consists of chlorophyll pigment, right? That is required for only photosynthesis, and photosynthesis happens only in plants. So it is present only in plants, not in animal cell. Okay, but when we are looking at the last option, vacuole. Okay, vacuole, minor vacuoles are present in animal cells, but not a bigger one. So major cell size that is occupied by the vacuole in case of plant cell. Okay, moving to the next question now. So here you can see. Okay, nucleus, this is nucleus, then mitochondria, cell membrane, this outer membrane, cytoplasm, that is the fluid which is present here. Next question, F1 particles or oxysome or elementary particles are present in endoplasmic reticulum, chloroplast, mitochondria, Golgi complex. Okay, let us look at the answer. The right answer is option C, mitochondria. Okay, F1 particles or oxysomes or elementary particles. So we can call it with the same different names here. And all of these are the one which are usually present in the inner wall of mitochondria. So if you look at the structure here, you can see the dotted structure that you can see here. All of these are oxysomes which are present in the inner wall of the mitochondria. We know that mitochondria is double membrane bounded organelle. So here is a one membrane as well as here innermost membrane. So here in the innermost membrane, there is an attachment of oxysome. This oxysome are also known as elementary particles or F1 particles. Okay, so this is the one, oxysomes are the one which is responsible for respiratory chain phosphorylation. Okay, because of the presence of oxysomes, in case of mitochondria of a cellular, respiratory cell phosphorylation happens. So why it has to happen? What is the function of this? So respiratory chain phosphorylation, it has to happen because it is the one which is responsible for the synthesis of ATP, ADP molecules. That is the energy molecules. Adenosine triphosphate molecule as well as adenosine diphosphate molecules, which releases energy that is required for our body. Okay. So what do you mean by this phosphorylation now? Phosphorylation is nothing but it is one of a metabolic pathway where there will be electron transfer from electron donors to the electron acceptors. Okay, there will be transformation of these electrons from donors to acceptors. Okay, they are in the metabolic pathway. So that is what we call it as phosphorylation. It is one of a metabolic pathway. Okay, moving to the next question now. Which of the following has the largest number of chromosomes? Human beings, pteridophytes, elephants, bryophytes. Okay, let us look at the answer. The right answer is option B, pteridophytes. Okay, pteridophytes or the organisms, that is one of a phylum, one of a group of organisms which has the largest number of chromosomes. Okay, here the ferns are the one which belongs to pteridophyte. It is one of a pteridophyte. Okay, so these pteridophytes, they usually have large number of chromosomes set in their body. For example, here we have one of a fern. Okay, it is a group of, it is one of a type of pteridophyte here, belongs to pteridophyte group. So that is Ophioglossum. Okay, Ophioglossum reticulatum. Ophioglossum reticulatum. It is an organism which has about 1440 chromosomes. That is approximately 720 pairs of the chromosomes are present in case of Ophioglossum reticulatum. Okay, so it is the highest chromosomal number which is known in all the lifespan. Okay, life forms. So whether we consider the animals or the plants here, so this is the one of the organism which has the most maximum number of chromosomes, that is 1440 here. Okay, usually pteridophytes are the group of organisms which has maximum number of chromosomes. Moving to the next question. When one gene controls 2 or more different characters simultaneously, the phenomenon is called apomixis. Okay, apomixis, pleiotropy, polyploidy, polyteny. Okay, let us look at the right answer. The right answer is option B, pleiotropy. Okay, pleiotropy. So when multiple traits, that is multiple different species of organisms are all controlled by one particular gene. A single gene can control all the multiple traits of organisms. And this process, this phenomenon, we call it as pleiotropy. And this effect which was produced by this function, we call it as pleiotrophic effect. Okay, but when we are looking at the rest of it, polyploidy. Okay, polyploidy is a condition where the individuals with more than 2 complete sets of chromosomes are present. Individuals with more than 2 complete set of chromosomes. So such organisms, we call it as polyploidy. Okay, here in pleiotropy, one single gene controls the multiple traits of organisms. So again, when we are looking at apomixis, here the reproductive process in plants without the fusion of gametes. So without the fusion of the gametes itself, reproduction happens. Gametes, we know that they are the male and female sex cells, right? Without the fusion of these gametes, sex cells, usually the reproduction happens. So that is here in the case of apomixis. When we are looking at the last, that is polyteny. Polyteny is a condition of chromosomes or the nucleus or the cell of an organism in which DNA is repeatedly replicated without separation. Okay, so there in the case of chromosomes or the nucleus or the cell of an organism, the DNA would have replicated multiple times without separating. So that is a condition, that is a phenomenon, we call it as polyteny. Moving to the next question. Which one of the following is considered as a cell within a cell? Ribosome, chloroplast, lysosome, Golgi complex. Okay, let us look at the right answer. The right answer is option B, chloroplast. Chloroplast is one of a cell organelle in case of plant cells, right? It is present only in plant cells because this is the one organelle which is responsible for photosynthesis process because it carries chlorophyll pigment within that. Okay, so this is one of a cell organelle which is considered as a cell within a cell because so this is semi-autonomous in nature, semi-autonomous organelle. What do you mean by semi-autonomous now? Semi-autonomous is the one which can synthesize their own DNA. Okay, the cell which consists of their own DNA then such organisms, such cells, organelles can be called as semi-autonomous organelles. Organelles are the one which are present in a cell. If this organelle is capable of synthesizing, or if they have their own DNA, then that can be considered as semi-autonomous. If they have their own DNA, definitely they can also synthesize their own ribosomes. Okay, so you are out of the given option here. So this chloroplast is the one which has its own DNA. Next question, which one among the following cell organelles is semi-permeable? Cell membrane, plasma membrane, cell wall, nucleus. Okay, let us look at the right answer here. The right answer is option D, nucleus. Okay, we know that semi-permeable, which means, so here the membrane which will allow only the selected compounds to pass through it. Okay, it is a membrane which allow only selected particles to pass through it. Such membrane can be called as semi-permeable. It will not allow all the molecules that enters there to pass through it. Only selected molecules, selected substances can pass through it. So such a membrane, we can call it as semi-permeable. And when we are looking at all the options that we have here, nucleus is the one which has a semi-permeable membrane, so that only selected particles can move. But when we are looking at the rest of the options, cell membrane, plasma membrane, cell wall, all of these are semi-permeable in nature. But here, all of these are not the cell organelles. Okay, so whatever we have, the organelle which is suspended there in the cytoplasm, okay, if it is suspended there in a cytoplasm of a cell, only that can be called as cell organelle, right? But here the cell membrane is the outer covering, plasma membrane, cell wall, all of these are the outer covering of the cell. But nucleus is the only one cell organelle that we have in the given options here, because it is suspended there in the cytoplasm of a cell. And it is also semi-permeable nature, right? Since it is a cell organelle, we can choose this option here. Okay, option D here. Moving to the next question. Xylem tissue is mainly concerned with the photosynthesis of plants, transport of water and mineral nutrients in plants, storage of prepared foods in plants, transport of enzymes in plants. Okay, let us look at the right answer. The right answer is option B. Okay, xylem, it is one of a vascular tissue which is present in plants. Okay, xylem and phloem. These two are the vascular tissues, vascular bundles which are present in case of plants. And these two are the one which is responsible for transport of water and as well as food. Okay, water, we know that roots of the plants will absorb that and it is then supplied to all the parts of the plant body. Basically, it has to carry the same towards the leaf because there is a site of photosynthesis, right? So to carry this water from the roots towards the leaves, here we have this xylem tissue. Okay, xylem tissue, which is unidirectional in nature. That is, it will always move from the lowermost layer to the topmost layer, the topmost part of a plant. That is only in one direction, because it has to carry the water from the soil towards the upper part of a plant body. Okay, the xylem tissue is the one which is responsible for the transportation of water and mineral nutrients into plants, in the plants. Okay, phloem tissue is responsible to carry the food which is synthesized there in the plant leaves to other parts of a plant. Okay, xylem, water and minerals, phloem, food. That you have to remember. Moving to the next question. The correct sequence of cell cycle is yes, G1, G2, M. Yes, M, G1, G2, G1, S, G2, M, M, G1, G2, yes. Okay, let us know what the right answer here. The right answer is option C. Okay, it is option C here. Option C, G1, yes, G2, yum. That is gap phase 1, synthesis phase, gap 2 phase, and mitosis phase. Okay, this is a cell division cycle. So here you can see gap 1 phase followed by synthesis phase, gap 2 phase, and then finally mitosis phase. Okay, so regularly in this process, in this different stage, it will usually occur. The cell division usually occurs in case of the cells. Okay, when we are looking at this G1 phase, so this is a phase which is completely metabolically active and it grows continuously. This phase grows continuously because it has to synthesize all the raw materials which is required for the further cell division. So it will be completely active. And the synthesis phase, as we have already discussed that, so it is a phase stage where the DNA synthesis, DNA replication takes place. Okay, DNA, if it is kept ready, so the further division, further necessary parts, necessary process required for the digestion, required for the division, cell division will take place. Okay, so gap 2 phase, this is a phase where the proteins are synthesized, and here in mitosis phase, the actual cell division occur. That is cytokinesis, division of parent cell into 2 daughter cells. Okay, moving to the next question now. Which organelle in the cell other than nucleus contain DNA? Centriole, Golgi apparatus, lysosome, mitochondria. Okay, let us look at the right answer. The right answer is option D, mitochondria. So in the previous question there, we have seen that about plant cell, that is chloroplast. In case of plant cell, it is the one which usually consists of their own DNA. But here in animal cells, usually this mitochondria is the one which will usually contain its own DNA. Okay, since it has its own DNA here in mitochondria. Okay, so it is also called as semi-autonomous organelle because it has its own DNA. Okay, if any organelle, if it has DNA of its own, then that can be considered as semi-autonomous organelle. Okay, and this mitochondria, the DNA which is present here in the mitochondria, it contributes about 1% of the total DNA of the cell, which usually consists of closed circular 50 and 60 S type of DNA. So there are different types of DNA, and the DNA which is present and possessed by the mitochondria is 50S as well as 60S. Okay, and since there is a presence of DNA, it can synthesize its own ribosomes, and these ribosomes are further utilized to synthesize protein. Okay, since it is semi-autonomous in nature, it can synthesize ribosomes. With the help of ribosomes, it can also synthesize their own proteins. So we can consider this mitochondria as protein synthesis machinery of the cell. Clear? Moving to the next question, next part here, the next options that we have here. Centrioles. So these are the set of microtubules. Centrioles has a major role during cell division. Okay, it forms microtubules so that all the chromosomal particles, chromosomes are held together with the help of microtubules. So Golgi apparatus, they are the complex of vesicles. Okay, they have a folded membrane which are involved in secretion. That is a major function of Golgi apparatus, right? It will secrete the vesicles, transport vesicles, which is required for intracellular transportation. So whatever the components that has to be transported in the cytoplasm, they are in the cell. So here, Golgi apparatus has a major role because it is the one which can synthesize vacuoles. Okay, it can synthesize vesicle, which is then used for the transportation as well as dispatchment. Okay, dispatching various components. And when we are looking at this lysosomes, they contain degradative enzymes. Okay, degradative enzymes are necessary to digest the cell itself if it is worn out. Okay, if there is any foreign particles or if there is any damage in the cell organelles, if the cell itself has damage, lysosome can dissolved, can digest its own cell. Okay, lysosome is one of a cell organelle. But if the cell is damaged or if there is any toxic substances in the cell, lysosome has a capability that it can digest its own cell. So we can consider this lysosome as suicide bags of the cell. Mitochondria is the powerhouse of the cell, whereas lysosomes are considered as suicide bags of the cell. Next question. Biological membranes are composed of 40% protein and 60% lipids, 50% proteins and 50% lipids, 70% proteins and 30% lipids, 60% proteins and 40% lipids. Okay, let us look at the right answer. The right answer is option D, 60% of proteins and 40% of lipids. So in this ratio, the composition of this biological membrane, that is the membrane bounded organelles, the membranes which are usually semi-permeable in nature, okay, the membrane of different organelles that we can see here, there will be a composition of proteins and lipids in this percentage respectively. Okay, moving to the next question. An individual whose blood type is B may in an emergency donate blood to a person whose blood type is B or A, AB or A or O, AB or B. Okay, let us look at the right answer here. The right answer is option D, AB or B. Okay, a person whose blood is type B, okay, B blood group, they can donate it to a person who has a B blood group or AB blood group. Okay, person with blood group can be donated the blood to person with blood group B or AB, because B can be donated it with B only, normally, generally, right? Person with the same type of, since it is the same type of blood group, person with B blood group can donate it to B, but they can also donate it to AB blood group also, because we know that AB blood group, it is universal acceptor, right? So universal acceptor. So AB blood group, a person with AB blood group can receive any of the different types of blood, whether it can be O, E, AB or B. Okay, all the different types of blood group can be received by AB person, a person with blood group AB. Okay, moving to the next question. Blood cells do not shrink in blood because blood is hypertonic, isotonic, equimolar, hypotonic. Okay, let us look at the right answer. The right answer is option B, isotonic. The blood cells are isotonic in nature. That is the reason usually the blood cells will not shrink there in the blood. So what is the reason? What is the meaning of this isotonic now? When the osmotic pressure, that is the movement of water, right? So usually here in the osmosis process, the water molecules will move from the higher concentration to lower concentration generally. But here when we are considering the blood here, so here it is isotonic in nature. And due to this reason, blood will not usually shrink. That is the pressure outside as well as inside the blood cells. Okay, that is in the blood as well as the blood cells here, the pressure, blood pressure remains the same. Okay, the concentration of water will remain the same both inside the blood cells as well as outside the blood cells. That is the reason it is isotonic in nature. It neither moves out nor gets into the cell. The water molecules will remain the same. Even if it is moving inside, okay, for example, if we have a solution here, so and if we have a blood cells here, if this is isotonic condition, the water molecules that is moving out of the blood cell and the quantity of the water molecules that is entering into the cell from the outside surrounding will always remain the same in case of isotonic solution. So the number of water molecules that is moving out and the number of water molecules that is entering inside the cell will always remain the same. So there will be no any change in the water molecules. Water concentration will remain the same. So that is the reason we call it as isotonic equilibrium. Clear? That is the reason blood will not even shrink there. It will remain as it is there in case of blood. Okay, the blood cells will remain there in the same case of as it was in the blood. But what happens if it was hypertonic? Okay, if the blood cells, if we place it in hypertonic solution, what actually will happen? Hypertonic solution, it is a condition where there will be higher solute concentration outside the cell. That is in the solution that we have taken. Okay, for example, if we have taken a beaker which is full of hypertonic solution, that is the solute particle will be more in this water. And if you place blood cell here, what actually will happen? It will start shrinking down because the water concentration inside the cell is more than that of its concentration outside the cell. So all the water molecules will tend to move out of the cell because we know that during osmosis process, water will always move from higher concentration to lower concentration. Since the water concentration inside the cell is more here, it will start moving towards the lower concentration, that is to surrounding in this direction. Okay, as and when there is no water in the cell, definitely the cell will shrink, right? So in case of hypertonic solution, it will shrink. Okay, but what happens if it is hypotonic solution? So the same condition, if we take it here, we are taking hypotonic solution, that is the water with very less solute concentration. And if you are placing blood cell here, so now the water concentration outside is more than that of the cell. So water molecules will tend to move inside the cell because we know that it is moving from higher concentration to lower concentration. It will move in this direction. So now the blood cell will occupy with more and more water molecules. As and when more and more water molecules enters the cell, definitely the cell has to swell up, right? So that is what will happen in hypotonic solution. Since it is isotonic solution, it remains the same. It will neither shrink nor swell. Okay, moving to the next question. In plants, which one of the following tissues is dead? Parenchyma, collenchyma, sclerenchyma, phloem. Okay, let us look at the right answer. The right answer is option C, sclerenchyma. Sclerenchyma is considered as the dead tissue in case of plants, right? All of these that we have here, all these are different types of plant tissues. Parenchyma, collenchyma, sclerenchyma, and phloem. Phloem is a vascular tissue which is responsible for to carry the food synthesized there in the leaf to different parts of a plant, right? Phloem is a vascular tissue which carries food. But this parenchyma is a soft tissue which is responsible for the softness of the plant. Collenchyma, it provides flexibility to the plant, right? Sclerenchyma is the one, it is a dead tissue which helps in the formation of bark of the plant. Okay, the sclerenchyma is also found in cortex, pith, hypodermis, that is the innermost layer, the below layer of the external part, epidermis. Okay, pulp of the fruits. So even in young leaves, which are usually living, which has the protoplasm within them. Okay, so in all of these conditions, in all of these cases, we can find this sclerenchyma. Okay, it is a dead tissue. So, but even when we are looking at this mature cells, there will be formation of secondary walls, right? If there is a tree here, and if there is a formation of secondary walls here, we can see the dried part, right? The outer part of this tree will be dried one because there will be presence of this dead tissue called sclerenchyma. Okay, the sclerenchyma is the one which is, even though if it is dead, it is necessary for a plant body. How? Because the sclerenchyma, even though it is dead, it can provide the mechanical support as well as strength and rigidity to the plants. Because of all of this reason, all these different tissues has their major role in case of plants growth. Got it? It is equally necessary here. Got it? Okay. So students, in this topic, in this class today, you have learned about, we have discussed most of these important questions which are completely related to the topic cell biology, genetics, as well as blood. I hope you have understood now, and thank you for attending the class.